Case example number 2 is from lentivirus. So here the case is that the virus is grown on HEC293 cells, and they are grown on a complex growth medium containing human serum albumin. So, and the objectives are again to quantify the different viral proteins from the lentivirus, determine the amount and ratio of different viral proteins. Also, this client asked us if we could look for the transgene. It's a T-cell receptor expressed in the humans, and they also ask if it's expressed in the HEC293 cells. And thirdly, what is the amount of residual proteins? So just looking at the data, if you look at the picture at the bottom of the figure, you can see the viral proteins. So where you have the Gag-Pol polyprotein and the Gag polyprotein. So the protein expression level from these two and the ratio between these proteins, they are critical for maturation of the virus and also critical for correct viral assembly. So as a manufacturer, you want to monitor that you have the right ratio of these proteins in your product. So, and as you can see the table above, we can quantify each of the viral proteins, and you can look at the amounts and compare them between different batches. We could also see that the T-cell receptor was expressed in small amounts for 3 of the products. On the table on the right-hand side, you see the total amount of viral proteins in nanograms per ml. So really, it gives a lot of details about the proteins in the product. We also looked at the host cell proteins from the HEC293 cells, and from the table here again, you can see a long list of different proteins in the different samples. We can also identify the proteins from the growth medium, and we knew that human serum albumin was added, so we identified this specifically. So that's the top protein on the list, so a lot of albumin in there, but we also see other human proteins in lower amounts. So you can use this to monitor your products for consistency.